Hello everyone. My name is Rao. I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today, we will continue with the next lecture of the chapter Respiration in Plants. So, till last lecture, we studied about a universal cycle that occurs in respiration, and that is glycolysis. This glycolysis, irrespective of type of respiration, happens in both aerobic and anaerobic respiration. And then we also saw that what are the pathways that are involved in the anaerobic respiration. Now, if we recall what happened, so we saw that till there, the glucose in the glycolysis, the glucose was getting converted into two molecules of pyruvate, right? So two molecules of pyruvate was formed from one molecule of glucose. Now, what is the fate of this pyruvate? So what happens that this is your cell, right? And in this cell, your glycolysis is taking place in the cytosol, right? Or cytoplasm. So here the glucose is getting converted into pyruvate. Now this pyruvate, it enters in an organelle that we know, and that this organelle is what? This is mitochondria, right? This is mitochondria. And where further processing of this pyruvate takes place. Now, what happens that if you remember, during this glucose to pyruvate conversion, there are certain products that were being formed. So what were those products? So it lead to the formation of ATP, and this ATP was through substrate level phosphorylation. Apart from this, it also lead to the formation of NADPH. So the NADH. So two molecules of NADH were also formed. Now, what happens to this NADH? So this NADH, along with this pyruvate, this NADH also enters into the mitochondria. Now, if you remember in the last lecture, I also told that the NADH produce also in later on gets converted into ATP. So if you think how it happens, so what happens is that this NADH also enters into the mitochondrial matrix. And this pyruvate, once it enters into this mitochondria, so here the pyruvate enters into the mitochondria, it gets converted into a compound known as acetyl-CoA. And here also, it leads to the production of, it utilizes NAD plus and leads to the production of NADH. So here we have NADH, this NADH. So what actually this accumulation of NADH is doing? Okay. And then this acetyl-CoA, it enters into the TCA cycle. We'll study about TCA cycle today. And here also the NADH are formed. Okay. So what is the purpose of formation of this NADH? Because everything that is being formed, it must be utilized somewhere for the production of energy. This is the overall goal of the respiration, right? So what is happening with this NADH? So what does NADH do? Once they get accumulated into the mitochondrial matrix, these NADH undergo oxidation. Okay. So this NADH, all of this NADH, they undergo oxidation to produce NAD+. Now, if they undergo oxidation, for this purpose, they will require what? They will require electron from somewhere. And these electrons are provided by the electron carriers that are present on this inner membrane of the mitochondria. So this is the inner membrane. This is the inner membrane of mitochondria. And these electrons are present here or are supplied from here to this NADH. So NADH receive this electron, and this carriers, those carry this electron are known as electron transfer system. So this together constitutes electron transfer system. Okay. Now, once it receives this electron, it undergoes oxidation, and then what happens? That this Again, now it will lead to the formation of this H plus, right? This H plus will be formed separately. Now this H plus, it moves and gets accumulated into the periplasmic space. So periplasmic space is what? This region that is between inner and outer mitochondrial membrane. So it gets accumulated into periplasmic space. Okay. And next thing what happens after this accumulation? So we know that mitochondria, this have this invasion like this kind of structure where certain enzymes are located. One type of enzyme that is known as F0 F1 particle. So this H plus, it enters through this F0 F1 particle, and when it enters, it leads to the ATP production. So this is how the ATP production, this NADH, in turns leads to the formation of ATP. This process we will study in more detail, but for now, remember that this pyruvate, whatever it is forming, and whatever NADH is forming during the metabolism of pyruvate or glucose, this leads to the contribution of ATP formation, and this ATP is forming by some pathway similar to this one. Okay. So, and this pyruvate, it gets converted into acetyl CoA, and then this acetyl CoA enters into the TCA cycle. Now, if you remember, we discussed about that in case of aerobic respiration, we had three things. We had first glycolysis. The second one was glycolysis. The second one was link reaction. Link reaction. Third is TCA cycle. And the fourth is ultimately ETS that will lead to the ATP production from NADH. Now we have studied about the glycolysis. The second one is the link reaction. And what? For the hint, this is the link reaction that is happening here. Now let's see this reaction in detail. So the link reaction, why it is called a link reaction? Because it is a bridge. It is a bridge between, if you see in the figure, it is this reaction is a bridge between what? Glycolysis. This process is a glycolysis. And this is TCA cycle, right? So link reaction is a bridge between glycolysis and TCA cycle. That's why it is known as link reaction. Now, this reaction also has various names, such as it is also known as gateway reaction. It is also known as gateway reaction or transition reaction, because here the transition is taking place, right, from the cytoplasm to mitochondrial matrix. So it is also known as transition reaction. Then this is also known as oxidative decarboxylation reaction. Oxidative decarboxylation reaction. Okay. Now, why it is known as oxidative decarboxylation reaction, we will see shortly. Now, what is the location of the link reaction? So the location of this reaction is where it occurs in mitochondrial matrix. It occurs in mitochondrial matrix. Okay, so this is this reason. This inner to this inner membrane of mitochondria is the matrix. Okay, so it occurs in the mitochondrial matrix. Now, what is the reaction? So here we know that pyruvate, so this pyruvate, it undergo a series of changes. It undergo series of changes, and it leads to the formation of molecule known as acetyl CoA. Okay, but during this process, what it does, it utilizes NAD plus. Okay, it utilizes NAD plus and gets converted into this NAD plus undergo reduction and form NADH, right? And this pyruvate is 3-carbon molecule. Okay, we have seen in glycolysis that it is a 3-carbon molecule. Now, but this acetyl CoA, it is a 2-carbon molecule. So it only comprises of 2 carbons. Okay. So which means that there is release of one carbon molecule, right? And that release takes place in in the form of carbon dioxide. Okay. So what we are seeing here, we are seeing that here this NAD plus it is undergoing reduction. And if you remember this reaction, that if something is written like this and this is getting formed, so what we can say that here this NAD plus is going reduction, undergoing reduction, right? Then this A to B conversion, it is an oxidation reaction, right? It is oxidation, right? So what is happening here? Here this NAD plus is getting reduced, which means this pyruvate is getting oxidized. So we have two components here. One is the removal of this carbon dioxide. That is known as decarboxylation. And second is oxidation of pyruvate to acetyl-CoA. And because of this reason, it is known as oxidative decarboxylation reaction. Okay. So here, what is happening? Carbon dioxide is getting removed and pyruvate is getting oxidized. And hence it is known as oxidative decarboxylation. Okay. Now, this reaction is catalyzed by an enzyme complex. So it's not a single enzyme, but like it is a complex that comprises of multiple things along with the enzyme. And this complex is known as pyruvate dehydrogenase. So dehydrogenase, like we know that it is used when NAD plus or NADH is used, right? So the enzyme that catalyzes this reaction is known as dehydrogenase complex. So it is a complex known as pyruvate dehydrogenase complex, or it is also written as PDC. Okay. Now, since it is a complex, like I said, so it along with the enzyme, it also contains multiple things. So what all things it comprises of? So it comprises of magnesium ion. Okay. It also comprises of TPP. So TPP is a cofactor. Okay. It is a cofactor that whose full form is thymine pyrophosphate. Thymine pyrophosphate. Okay. So it is a cofactor. It is a cofactor. Now, the third thing that is consists of is an acid that is known as lipoic acid. Lipoic acid. And the fourth thing it has is this one, that is NAD plus. NAD plus. Okay. So these are the all things that is present in this pyruvate dehydrogenase complex. Okay. Now, in the NCERT, the reaction is written this reaction only. It is written in the form something like this. So you have pyruvate. Here, this NAD plus is written in the substrate because if you look, it is anyway a substrate. And it also have coenzyme A. So a coenzyme A that I should write here like this. Coenzyme A, it is also represented as CoA. Okay. So in the presence of this, what is happening? It is getting converted into acetyl-CoA. Then another product is what? CO2, right? And another product is NADH. NADH plus H plus, right? H plus. So this is what is the total complete reaction. And the complex that catalyzes PDC and Mg2 plus. So it comprises of almost everything. Now, yes. So you have this coenzyme. So what things we are seeing? So we said that we have seen that it has this pyruvate is undergoing decarboxylation because it is releasing carbon dioxide. It is undergoing oxidation because NAD plus here is getting reduced. And the third thing is this coenzyme A, what it does, it performs that function of acetylation. Okay. So this acetyl-CoA that is forming, which means this CoA is undergoing acetylation. So this is what? CoA. When you add an acetyl group here, it will become acetyl-CoA. And this process is known as acetylation. So if we talk about the function of this PDC, so function of PDC, that is pyruvate dehydrogenase complex, what all function we have? So it performs three functions. The first function is decarboxylation, right? The first function is decarboxylation. Now, what is the second function? The second function is oxidation, right? Oxidation, because oxidizing pyruvate. And the third function is what? Acetylation. Acetylation, because what is happening here, this coenzyme A, that is a substrate, it is undergoing acetylation to form acetyl CoA. Okay, so this is the function of this PDC. Now, we know that from one glucose molecule, how many molecules of pyruvate are formed? 2 molecules, right? So it means that this link reaction will take place from one glucose, it will take place 2 times. So this NAD plus, it will utilize 2 NAD plus to form 2 NADH. It will release 2 carbon dioxide if we are talking in terms of one glucose, and it will lead to the synthesis of 2 acetyl CoA, right? So this is the overall stoichiometry if we use one glucose. So what we can write? So we can write that in link reaction, in terms of link reaction, if you say in link reaction, what thing we have? If we are talking about one pyruvate molecule, okay, if we are talking just in terms of pyruvate, so from one pyruvate molecule, how many number of carbon dioxides are produced? So number of CO2 from one pyruvate is what? 1, right? And number of NADH producer? Number of NADH produced. This is also 1, right? If there's one pyruvate. But if I'm saying that from one glucose, if the question is like from one glucose, how many numbers of carbon dioxide and NADH is formed? So number of carbon dioxide will be 2, because from one glucose, 2 pyruvate is being formed. And also number of NADH that is produced is also 2. Okay. And number of acetyl CoA here, the number of acetyl CoA, it is 1, but in the second case, from one glucose, it is 2. Okay. And this is specifically in terms of link reaction. Okay. It does not include glycolysis. If it will ask about the glycolysis, we will add the things that we have studied in the last class, right? Okay. Now, so we have completed glycolysis, we have completed link reaction. Now the third is TCA cycle. Okay. So the TCA cycle, it is also known as Krebs cycle. Okay. So it has multiple names based on what product it is forming. It has, or what is its function, it has multiple. So the Krebs cycle, because it was discovered by the scientist Krebs, so because of this, it is known as Krebs cycle. But apart from this, it is also known as citric acid cycle. It is also known as citric acid cycle. Now, why it is known as citric acid cycle? Because citric acid is the first product to be formed in this cycle. Okay, so it is the first product to be formed, and because of this, it is known as citric acid cycle. Now, the second is, it is also known as tricarboxylic acid cycle. Tricarboxylic or TCA cycle. Okay. Tricarboxylic acid cycle, or you can also say TCA cycle. Now, why it is known as TCA cycle? Because if you look at the structure of the citric acid, it comprises of 3 carboxyl group. So it is known as this because citric acid consists 3 COH group, 3 carboxylic acid groups. Okay. And because of this, it is known as TCA cycle or tricarboxylic acid cycle. And citric acid, why we are considering? Because this is the first product to be formed in the Krebs cycle. Okay. And why it is known as Krebs cycle? Because it is named after the scientist. Named after the scientist who discovered this pathway. Okay. Now, what is the location of TCA cycle? So the location, it is same as the link reaction, that is mitochondrial matrix. It is located in the mitochondrial matrix. Okay. Okay. Now, how many steps are involved in this? So total steps that are involved in this is 10. We'll see. So this is a typical TCA cycle, but we will understand each of these things one by one, that what is forming, what are the enzymes involved. Okay. And then the number of enzymes that are involved. So the number of enzymes, although this number of steps are 10, the number of enzymes that are involved is 9. Okay. So we'll see that there is a repetition of one enzyme. Okay. Now, what is the use of this Krebs cycle? So if you see what we said about the respiration, so in that respiration, what happened? The glucose gets converted into carbon dioxide and the energy is formed, right? So this Krebs cycle is the place where this carbon dioxide that is formed, the glucose that leads to the formation of carbon dioxide, it gets completely utilized or gets completely released. Okay. So it leads to the, you can say, it leads to the complete oxidation. Here, the complete oxidation of glucose takes place. Complete oxidation of glucose takes place. Okay. And by complete oxidation, what it means? It means that the glucose finally gets converted into 6 molecule of carbon dioxide. Right. Now, this glucose, it is not getting entered into the TCA cycle directly, right? So this glucose, now it will enter into the TCA cycle in the form of acetyl-CoA, right? Because from glucose, ultimately the acetyl-CoA is formed. And this acetyl-CoA here, it enters into this TCA cycle. Okay. Okay. Now we will see how these products are formed. But for now, what are the products? Sorry. Just give me a second. Okay. So we will see that what are the products that are, how these products are formed. But for now, just remember the products that are formed are carbon dioxide, ATP, and this ATP is through substrate level phosphorylation. It forms through substrate level phosphorylation. Then you will have NADH and FADH2. So this is something new we have seen. So, but we'll see how it forms. And these two are produce ATP indirectly. Produce ATP indirectly. And in the first lecture of the slide, we saw that how these produces ATP indirectly. Okay, so NADH we saw, but FADH2 also produces energy in the similar ATP in the similar manner. Okay, okay. Now, so this is about the Krebs cycle, and this cycle, because it is known as cycle because you can see this is the structure. So, okay, so it is in the cyclic form. And here we are seeing this reaction here is, this is what? This is your link reaction. This is the link reaction. And from this link reaction, this acetyl-CoA, it enters into the TCA cycle. Okay. Now we'll study these steps in the TCA cycle one by one. So the first step of the TCA cycle is known as condensation reaction. What is the first step? Step 1 is the condensation reaction. Condensation reaction. Now, what does condensation mean? It means that we have two molecules, they come and combine with each other to form a single molecule. Okay, and there is release of nothing. Okay, so the two reactors, they completely get used up to form a single compound. So this is what condensation reaction in simple terms. In chemistry, you will see it in more detail. So what is happening here? So if you see the cycle, here we have acetyl-CoA coming. Here we are seeing another molecule, that is what? Oxaloacetic acid. Okay. So in the mitochondrial matrix, oxaloacetic acid is present. And this oxaloacetic acid acetic acid. This oxaloacetic acid is of how many carbons? So it is a 4 carbon molecule. Okay, it is a 4 carbon molecule. This will combine with the acetyl CoA that is coming from where? It is coming from the link reaction. And then it gets converted into, and this acetyl CoA is of 2 carbon, right? And then it gets converted into the first product of the Krebs cycle, which is citric acid. So it gets converted into citric acid. Okay. Now this citric acid will be is of how many carbon? It is of 6 carbon. So this 4 carbon and this 2 carbon combine to form the 6 carbon molecule. Okay. Now it utilizes something. Okay. So this utilizes this utilizes water molecule. Okay. So this condensation reaction, it utilizes water molecule to form this. And what is the enzyme that is involved that catalyzes this reaction? So the enzyme is known as citrate synthase. Okay, the enzyme is known as citrate synthase. You can remember it as the enzyme that is synthesizing the citrate, and hence it is known as citrate synthase. Now, so in this we have utilized a 4 carbon and 2 carbon molecule to form a 6 carbon molecule. Now we have step 2. What do we have in the step 2? So in step 2, the citric acid that is formed, it gets converted into an unstable product. Okay, that is not mentioned in NCERT, but this intermediate product is being formed. That is highly unstable, and this product is known as aconitic acid. Aconitic acid. Okay, and this also, what happens in this case? The water molecule that was taken in the step 1, it gets released. Okay, so the aconitic acid is formed when the water molecule gets released. That is utilized in the first step. And the enzyme that catalyzes this reaction is known as aconitase. Okay, so this is the enzyme that catalyzes this reaction. Still, it is 6 carbon, and here there is just removal of H2, so it will be 6 carbon. Okay. Now let's move to the third step. So what do we have in the third step? So step 3, what is happening? This aconitic acid, like I told, it is highly unstable, right? So this acid is highly unstable. So it instantaneously gets converted into another product that is known as isocitrate. So it gets converted into isocitrate. And how it gets converted? It take this water molecule again. Okay. So this step, it is what? It is unstable. This is unstable. And this step takes place for a very small scale of time. Okay. Now, this enzyme that catalyzes this reaction is again the same enzyme, that is aconitase. Okay. So this aconitase, it within a very short span of time, it performs 2 reactions. First of all, it converts the citric acid into an intermediate. Okay. So you can also think it as an intermediate. Aconitic acid as an intermediate. Okay. So this intermediate that is highly unstable. And again, this enzyme further converts this aconitic acid into isocitrate. Still, there is no release of any carbon molecule. So it is 6 carbon, right? But what we are saying is that the ultimate goal is to release the carbon dioxide, right? This is how glucose oxidation takes place. So what happens next? Now in the step 4, what happens? This isocitrate that is formed, it gets converted into oxalosuccinate. Okay. Until now, there is no ATP formation taking place, no carbon dioxide release took place. So here it is still 6 carbon and 6 carbon. but just the rearrangement of these molecules are taking place, leading to the formation of different products. Okay. Here what is happening? Now, here, if you see, here comes the utilization of NAD plus. So this isocitrate, in the presence of NAD plus, it gets converted into oxalosuccinate. So here what is happening? This NAD plus is getting reduced and this isocitrate is getting oxidized to form oxalosuccinate. So this is what? Oxidized, or you can say oxidation. So there is oxidation of isocitrate to oxalosuccinate. And now we can guess that what enzymes might have involved. So we can see here that now NAD plus is involved, right? And whenever there is NAD plus or NADH, what enzyme catalyzes this reaction? So the enzyme that catalyzes this reaction is known as isocitrate dehydrogenase. Okay, so the enzyme will be dehydrogenase. That is easy to remember. And because it is catalyzing the reaction involving a substrate named isocitrate, it is known as isocitrate dehydrogenase. Okay, but the thing is, although there is an oxidation reaction, but still we have 6-carbon molecule. Okay, still no carbon dioxide is released. Okay, now what do we have in step 5? In step 5, this oxaloacetate, it gets converted into a compound known as alpha ketoglutarate. Alpha ketoglutarate. Okay. This oxaloacetate is a 6-carbon, we know, but this alpha ketoglutarate, it is a 5-carbon molecule. So now we have finally a release of one carbon dioxide molecule, right? So here what is happening? It is releasing the carbon dioxide. Okay. And if an enzyme that releases the carbon dioxide, then what that enzyme is known as? That enzyme is known as decarboxylase, right? So this enzyme is known as decarboxylase. Okay. It is known as decarboxylase. Oxaloacetate decarboxylase enzyme catalyzes this oxaloacetate. If you see the acetyl-CoA that enter here, this acetyl-CoA, it has 2 carbons, right? So the goal should be that in one TCA cycle, 2 carbon dioxide should get released, right? So that brings us to the next step. So what is the next step? So in the next step, this alpha ketoglutarate that is formed. Okay, this alpha ketoglutarate. Yeah. So in step 6, the alpha ketoglutarate that is formed, or you can also call it as alpha ketoglutaric acid. That gets converted into another compound known as succinyl coenzyme A. So it is known as succinyl coenzyme A that I am representing by CoA. Okay. Now, just guess that if this is alpha ketoglutarate, it do not have any coenzyme, but the product that is formed comprises of coenzyme A. So it means that this step requires the coenzyme, right? So here, what happens? The coenzyme comes into picture. So here, the coenzyme A addition takes place, and hence this reaction you can also call this as acetylation reaction, right? Now what happens? Here, the coenzyme A came, and along with this, you have release of, because this alpha ketoglutarate, it is a 5-carbon molecule, but this succinyl CoA, it's a 4-carbon molecule, which means the coenzyme A enters, but there's release of carbon dioxide. So till this step, the carbon dioxide that, like the 2 carbon dioxide that should be released by the addition of acetyl CoA, it has happened. Okay, so 2 carbon dioxide molecules have been released till step 6. Okay. Okay, so here the coenzyme A enters CO2, and apart from this, it also, at this step, one more thing happened, and this thing is what? This NAD plus gets converted into NADH. Okay, so the NADH is formed. Now, we have multiple things happening here. If you recall, we have seen this kind of reaction previously as well, right? So where the coenzyme addition is taking place, carbon dioxide release is taking place, and NADS is getting converted into NADH. And what is this reaction? This reaction is your link reaction, right? So it is looking something like this, right? Where NADH is forming, carbon dioxide is getting released, and coenzyme is getting added. And this is catalyzed by an what do you call enzyme whose name is also similar to the previous enzyme, and this is known as alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase, right? So this enzyme catalyzes this reaction where the 5 carbon molecule is getting converted into 4 carbon molecule, and finally the carbon dioxide that enters in the form of acetyl CoA, it gets released. Okay, so this is the step 6. Now, what do we have in step 7? So, like I said, there are 10 steps, right? Now, in the step 7, what happens? The succinyl CoA, it gets converted into succinic acid. Now, no, because the carbon dioxide that should be released has got released. The acetyl CoA comprises of 2 carbon molecules, and 2 carbon dioxide are released. So, from this step onward, there will be no reduction of number of carbon molecules in the products. Okay. So this succinyl CoA, it gets converted into succinic acid. It gets converted into acid known as succinic acid, or it is also known as succinate. Okay. And here what happens? Now, why this cycle should proceed? The carbon dioxide is formed. You can say that glucose is oxidized, but we still need energy, right? And till now, there is no ATP production. But at this step, at step 7, if you see, so the succinyl CoA, it gets converted into succinic acid. And what happens here? It happens by the utilization of a water molecule, release. So this is the utilization. The arrow here is the release of what? Coenzyme, because here the succinic acid is there. Here this coenzyme is got released. And apart from this, what happens? This happens. The GDP that is present in the mitochondrial matrix in the presence of phosphate ion, it gets converted into GTP. So just like ATP, GTP is also a form of energy. Okay. That ATP, what do we have? We have adenosine triphosphate. GTP, what do we have? We call it as guanosine triphosphate. Okay. So it utilizes the phosphate ion and gets converted into GTP. And this step here is known as substrate level phosphorylation. So substrate level phosphorylation is something that it is not happening through the use of F0 F1 particles. So we'll see in, I guess, tomorrow's lecture that this phosphate, so this energy is mainly produced in the cell via this F0 F1 particle, but there are some energy that are produced during this substrate level phosphorylation, where the phosphate is taken up from the substrate or from anywhere in the cell, and then it gets converted into this GTP or ATP. And this is known as substrate level phosphorylation. Now, what we have seen that in step 7, that this succinyl CoA, it gets converted into succinic acid, and the enzyme that catalyzes this reaction is known as. So whenever there is an involvement of phosphate group, we should remember 2 things. If there is involvement of phosphate, then it is known as, the enzyme that catalyzes that is known as kinase. Okay. And if there is involvement of second thing, that is NAD plus or NADH, then the enzyme that catalyzes those reactions are known as dehydrogenase. So it will make life a little bit simpler if we can remember this thing. So if we are seeing that here the GTP formation is taking place, there is phosphate group involved. So the enzyme must be kinase. And then we'll have to take a wild guess that what can be the complete name. So since we are seeing that it's succinyl CoA, the enzyme name should be something like succinate. And it also involves, this is a special case where it involves this thiokinase. Okay. So the enzyme that catalyzes this reaction is succinate thiokinase. So till now we have completed how many? Step 7. Now what is the next step? So the next step is step 8. So what happens in the step 8? Just a second. Okay. So what happens in step 8? In step 8, this succinate or succinic acid that is formed, the succinic acid that is formed, this succinic acid, it gets converted into another compound that is known as fumarate. Okay. And it utilizes FAD. So like I said in the first slide of related to Krebs cycle, that we will see another coenzyme that is FAD. And this FAD undergo reduction to form FADH2. So it is just like NADH. What is the difference? The difference is just that NADH leads to the production of higher amount of ATP as compared to FAD. But both are like similar. Okay. So this succinic acid, it gets converted into fumarate enzyme, fumarate compound, and the enzyme that catalyzes this reaction. So here again, if it is FADH, so not only in the case of NAD, but if FADH is also involved, in that case also the enzyme will be dehydrogenase. Okay, so the enzyme will be, because it is a succinic acid, or it can also be called as succinate, the enzyme will be succinate dehydrogenase. Succinate dehydrogenase. Okay, so this is the 8th step. Now, there's one thing interesting about the succinate dehydrogenase, and we have also studied in biomolecule chapters, which is the succinate dehydrogenase. If we talk about that, I'm representing by ST. The substrate for this enzyme is succinic acid, right? So its substrate is what? Succinic acid. But it has an inhibitor. This enzyme, we know the enzymes do have inhibitors, right? They can have inhibitors. So the inhibitor of this enzyme is malonate. Malonate. And if you remember, what is the similarity between this succinate or succinic acid and malonate or malonic acid is they have a similar kind of structure. Okay. So there was, I think, one methyl group difference. So the inhibition that takes place, this is the inhibitor, and it inhibits this enzyme, succinate dehydrogenase, by competitive inhibition. By competitive inhibition, because it has a structure similar to that of this enzyme substrate, that is succinic acid. Okay. So this is about the succinate dehydrogenase. Now, the next is, we have completed how many steps? We have completed 8 steps. Now, the next step is step 9. And what do we have in step 9? So in step 9, this step 9, our fumarate, it gets converted into malic acid. It gets converted into malic acid. Okay. And all of these are how many carbon molecule? So from succinyl-CoA till here, everything is 4 carbon molecule. Okay, it is 4 carbon molecule. And this fumarate to malic acid takes place by the hydration reaction, that is S2 chem, and catalyzed by the enzyme known as fumarase. Okay, it is catalyzed by enzyme known as fumarase. And this is what? Hydration, right? H2O is getting added. Okay. And finally, the step 10. Step 10. So what happens in step 10? Here, the malic acid finally gets converted into what? The initial, from where we started, that was oxaloacetic acid. Oxaloacetic acid. So this is 4 carbon, and the acetic acid, if you remember, that was 4 carbon, right? So again, the oxaloacetic acid got recovered. Okay. And this was, here again, what is happening? The NAD plus is getting utilized to form NADH. It means that a malic acid undergo oxidation to form oxaloacetate. And here the enzyme that is involved is known as malate dehydrogenase. Where whenever there is NADH, the enzyme will be dehydrogenase, right? So this is what the complete cycle includes. So now we have understood why it is known as cycle, because ultimately we started with the oxaloacetic acid and we came back to the oxaloacetic acid. So it is taking place in a cycle, right? Okay. Now, what is the important point? The important point is like where this GTP is produced, where this substrate level phosphorylation is happening, where there is NADH production, where there is FADH2 production. So there is a lot of things to remember. So just to summarize these things, what we can say. So let's write that how many number of decarboxylation steps are here? Number of decarboxylation steps. So we understand what is decarboxylation, right? Decarboxylation is removal of carbon dioxide. So how many steps are there? So we started with acetyl CoA. Acetyl CoA is two carbon molecule. So there should be two release of carbon dioxide. And hence, and we can also see this from the reaction. There is one step here, right? And there is another step here. So the number of decarboxylations are what? How many? Two. And what are the steps? At which steps it is happening? So the steps that involve is fifth and sixth step. Fifth and sixth steps. Okay. At these two steps, decarboxylation is happening. Now, how many number of NADH formed? So I'm talking about one acetyl CoA molecule. Just remember this, from one acetyl CoA. This is from one acetyl CoA. Okay. So now, how many NADH will be formed? So the NADH that will be formed will be 3. Okay. We can see here there is one NADH, there is second NADH, and then this is third NADH. So number of NADH is 3. And what are the steps at which this NADH formation takes place? So it takes place at 4th, 6th, and 10th step. So it takes place at 4th, 6th, and 10th step. Okay. Now, what is the number of FADH2 formed? So FADH2 we saw just once, right here. So FADH2 is formed is 1, and the step is which step? 8th step. 8th step. Right? Okay. Now, about the hydration. So where the hydration takes place? Hydration means addition of water molecule. So the hydration takes place at here at the 9th step, and where at the 7th step here also the hydration is taking place. Then we have hydration here and the first step. So we have first, third, 7th, and 10th. 9th, sorry. So hydration 4 places, and what are the steps? Steps are first, third, 7th, and 9th step. Okay. Okay. So this is about the hydration. Now, one important thing, number of substrate level phosphorylation, the number of SLP. So what are the number of SLPs that are formed? Substrate level phosphorylation is happening only once at step 7, and the GTP that is formed is 1. So number is 1, and the step is what? 7th. Okay. So this is the summary of what we have seen. Okay. And you should also practice this cycle. So the cycle in this, the complete information is not given, but what are the intermediates where the carbon dioxide is getting formed, how it is getting released, that thing we can see here. Okay. Okay. So this thing, what I told, that this is from one acetyl CoA, right? But what we have seen that from one pyruvate molecule, we have, from one glucose molecule, we have 2 acetyl CoA forms, right? So what is happening? What happens is from one glucose, it leads to the formation of 2 pyruvate, right? And 2 pyruvate leads to the formation of 2 acetyl CoA, right? 2 acetyl CoA. So for one glucose molecule, how many times this TCA cycle will run? So for one glucose, 2 times this TCA will run, right? for first time for one acetyl CoA and second for second. So for one glucose, TCA cycle will run 2 times, right? 2 times. So in that case, if the question is that this question is from one acetyl CoA, but if the question is that what will happen in the case of one glucose molecule, then all of this number will double, right? So for one glucose, if something is asked, then all of this number will be doubled. Okay. So the number of decarboxylation will be 4, number of NADH will be 3, number of peptides will be 2, hydration 8, right? So number of substrate level phosphorylation 2. Okay. So this is for one glucose molecule. Now, so this is about the Krebs cycle. Now, the one important thing about balance sheet of ATP. So with this, we have completed the aerobic respiration. Not completed. We have just one step left, that is ETS. But before that, let's see that from one glucose molecule, what things do we have? How many energy? What energy do we have? Okay. So this is called as balance sheet of ATP. So what happens in the balance sheet of ATP? So just to summarize, let's quickly write that 1 ton of Krebs cycle. 1 ton of Krebs cycle. What do we have? We have 3 NADH, right? 3 NADH, 1 FADH, 2, 1 FADS2, and 2 CO2. 2 CO2, right? And GTP, how many we have? We have 1. Okay. This is from 1 ton of Krebs cycle. Now, for one glucose, what we'll have? We'll have 6 NADH, 2 FADS2, 4 CO2, and 2 GTP, right? So this is for Krebs cycle, like in the Krebs cycle. So from one Krebs cycle, it will happen, but for one glucose in a Krebs cycle, this thing will be produced. Okay. Now let's see from starting from the glycolysis till the Krebs cycle, how many ATPs are produced. So we can write that phase is source and then ATP. Okay, ATP. So we have, what are the phases do we have? So we have till now we have studied about the 3 phases. The first one is what? Glycolysis. Glycolysis. Now in the glycolysis, we have what are the sources of energy or ATP? So we have one substrate level phosphorylation. One source is substrate level phosphorylation. And from substrate level phosphorylation, how many ATPs were produced? So we saw the net ATP production was 2 ATP, right? So it will go directly here to form 2 ATP. Now in the glycolysis itself, the second source was NADH. So how many NADH were produced during glycolysis? So the NADH was also 2. And from 2 NADH, so just remember, from 1 NADH, we can produce 3 ATP. Okay, we can produce 3 ATP. And from 1 FADS2, how we will see, but from 1 FADS2, we can produce 2 ATP. So this is the thing you should remember. Okay, so from 2 NADH, how many ATP will we produce? 6 ATP. But the first case is what? In this substrate level phosphorylation, this is what? This is direct ATP, right? So this one is direct, but this is indirect. It will go through the ETS system and then it will produce. So it is what? Indirect. Okay. So the first phase is glycolysis phase. Now, what is the second phase? It is link reaction. It is link reaction. Now in link reaction, is there any substrate level phosphorylation? No. So the substrate level phosphorylation is from there is no ATP. How many NADH are produced? From one glucose, this thing is. So NADPH that is produced is 2. So we have 2 NADH, and this 2 NADH will ultimately go and produce 6 ATP. Okay, so this is the second link reaction. Now the third is what we have is Krebs cycle that we just studied. The third one is Krebs cycle. Now in Krebs cycle, we know the substrate level phosphorylation. From substrate level phosphorylation, we produce 1 GTP, right? So 1 GTP, it will go here. You can either write GTP or ATP. Okay, we have 1. And then how many FADH2 we have? We have how many NADH we have? We have 6 NADH. 6 NADH. And this 6 NADH will go and produce 18 ATP. Then we have 2 FADH2. Right? We have 2 FADH2. And this 2 FADH2, it will produce what? It will produce 2 to the 4 ATP. Out of this, these two are what? Indirect. Because here NADH and FADH2 will indirectly contribute to an ATP synthesis, and this one is what? Direct. Similarly, this one is what? Indirect. Okay. This is also indirect. This is direct. Okay. So this will also help you remember that how many ATPs are produced directly and how many indirectly. By directly, what I mean? I mean via substrate level phosphorylation. Okay. So ultimately, the total ATP that will be produced at the end of one glucose consumption during aerobic respiration will be how much? So it's 1 here. Sorry, it will be 2 now. It will be 2 because we are talking in the terms of glucose. So here it will be 2. Okay. So how many will be? 6 to 8, 8, 6, 40, 60, 20, 38. So we'll have total of 38 ATP will be produced if one glucose molecule is consumed or utilized during aerobic respiration. During aerobic respiration. So in the aerobic respiration only, this linked reaction in Krebs cycle happens, right? So during aerobic reaction, 38 ATPs are formed if we take one glucose molecule. Okay, so this is the balance sheet. And the important points to remember is that what are the direct form of ATP energy and what are the indirect form. So if we talk about the direct energy ATP that is formed, how many direct ATPs are formed? So here is 2 and here is 2, right? Only 4. And rest are indirect, that is through the F0, F1 particle and electron transport system. Okay. So with this, we have completed the Krebs cycle. In tomorrow's lecture, in next lecture, we'll study about the electron transport system or oxidative phosphorylation. So that's all for today. Thank you.